I'm very glad to have Dr. Amber Smith with us today, and is a social professor in the Department of Pediatrics and the Institute for the Study of Host Passenger Systems at the University of Toronto Health Science Center, or UTHSC, in the beautiful city of Memphis. And Amber is tremendously well known in the community of mathematical epidemiology, especially in terms of the interface of the immunology and epidemiology, and she is a very active member in the International Society for Mathematical Biology, was an active member of the board of directors. So it's consecutive time in 1996 and 2016 and 2020, and she is also in the SMB's Immunology and Infection Subgroup Advising Committee. So I'm very pleased to have Amber to speak in the Fields Institute and in this particular colloquium. So Dr. Smith, please.
Thank you so much, and thank you for that introduction. I'll get my slides up. All right. So today I'm going to talk about a couple different pathogens that are all of significant public health importance. One we heard about a lot over the last several years, which is SARS-CoV-2, and one is starting to pop back up, which is influenza. And then the second part of both of these is looking at bacterial co-infections that occur with these virus infections. So most of the work I'm going to be talking about today was done within my lab. A lot of the data was collected by my very talented lab manager, Amanda Smith. And then the modeling was done by myself and a few graduate students and postdocs within my lab. The SARS-CoV-2 work was done in collaboration with another lab here in Memphis. That's Colleen Johnson's lab. She's the director of our institute and of our regional biocontainment lab, who helped a lot with these studies. And then our collaborators in the Channapanavar lab at Oklahoma State University. So as you're probably all very much aware about SARS-CoV-2, we've heard a lot about this over the last couple of years for obvious reasons. SARS-CoV-2 is an RNA virus that it mostly infects the respiratory tract, although it infects lots of other tissues as well. In the US, we've had an estimated 98 million infections and approximately 1 million deaths so far. In Canada, this has been significantly less, probably because there's a lot better public health measures that were undertaken during the pandemic. And the estimated numbers are about 4.4 million infections and 48,000 deaths. So as you're also probably aware, there's vaccines that are available. They seem to be pretty well effective, although the virus is mutating, and a couple updates have been made. But there are some reports coming out that they might be a little bit problematic, in the sense that there's, a handful of people that are developing autoimmunity, or maybe even a little bit of toxicity of those vaccines. So that what I'm going to talk a lot about today are the secondary bacterial infections. We haven't actually heard a lot about this with SARS-CoV-2, and that's probably in part due to poor reporting, but I'll get to that towards the end of my talk. And you can see these epidemic curves. This data is from the US because we had significantly more infections where we thought 2020 was bad. 2021 got a little bit worse. And of course, at the end of 2020 is when we saw a significant increase in the infections. And now it looks like we're maybe probably in much more of a seasonal, or maybe even a bi-yearly type of epidemic curve. So influenza, you might not have heard about over the last couple years because it was almost non-existent during the COVID pandemic. That's probably for a handful of reasons. One that I think probably drove a lot of it was that children were largely at home and not in school. and influenza affects children quite a bit more than COVID does. But it's also an RNA virus that infects the respiratory tract. In the US, there's usually 10 to 30 million infections every year and about 20,000 deaths. In Canada, I couldn't find a solid number for an approximate number of infections, but there's about 3,500 deaths, which thankfully is much less than what it is here in the United States. So unlike COVID, secondary bacterial infections have long been known to affect influenza and increase both morbidity and mortality of influenza. This is during a pandemic year, 40 to 95% of that mortality is really from the secondary bacterial infection and not so much from the virus infection itself. There are vaccines available. They have to get updated every year because influenza mutates quite a bit. And there's lots of reasons why there's numerous strains of influenza, whereas we don't see that with COVIDs quite as much. There are also antiviral therapies, but you have to give them very early on within, say, the first 48 hours for them to be most effective. And so because that doesn't always happen, we see a lot of infections. So if you look at these epidemic curves, these are dating back to the 2017-18 season. Pretty much every year, about the same time, we see a good epidemic curve of influenza. In this magenta line on the bottom, that's the 2020-2021 season. And then in the orange, where we did see a little peak, but for the most part was pretty low, that's the 2021-22 season. But over here in the red, we're already at a significant increase in influenza-like illnesses for this season. And if you probably haven't heard about it in the news, lots of viruses are sort of coming back with a vengeance. Influenza is one of them. RSV is one of them, where we're seeing a lot of infections. And so this season's not looking great for us so far. If we look at the number of pediatric deaths, because the pediatric population is one that is highly affected by influenza, usually we see, a couple hundred pediatric deaths reported. 2020 to 2021, there was only one. So this is maybe the silver lining of the pandemic, is that we didn't have quite as much mortality or even morbidity associated with other virus infections. But now that we're starting to see more, of course, these are starting to increase, and it's probably based on the season so far, it's probably not going to look good for 2022 and 2023. So both of these infections are really highly inflammatory diseases. They, like a lot of other viruses, have very similar viral load kinetics in the sense that you get rapid viral growth initially, and then maybe more of a slow decline as the immune response is kicking up, and your adaptive immune responses are coming in to clear out that virus. One of the big differences between these two infections is the time scale. So influenza is a very rapid infection compared to COVID, where it's peaked maybe by day 2, and by about day 7 to 10, you've completely cleared the infection. But with COVID, it maybe peaks more around day 5 and takes a couple weeks to kind of clear out and for people to start feeling better. These two images are CT images from severely infected patients. And what you can see here is the lighter shaded areas are where the damage of the virus was. And so you can see that on both sides of the lung. You can see it for SARS-CoV-2. Both sides of the lung have a significant portion of the lung that becomes infected or at least damaged by having the virus infection there. And this is probably in part due to some dysregulated immune responses. So of course, your immune response is supposed to get initiated, say with interferons and macrophages and neutrophils, and your innate immune response should come in and take care of and slow down the virus replication to allow time for your adaptive response, which is more of your CD8 T cells, to come in and clear that out. But during both of these infections, and to different levels, we see really dysfunctional types of immune responses, either lots of macrophages and T cells or too few, lots of cytokines coming in, and then we ultimately end up in a situation of having pneumonia or having widespread inflammation. And with COVID, and now we're starting to understand with influenza, there might be multi-organs that are affected and maybe even damaged by the virus. For bacterial co-infections, there's been a lot of work done on looking at these with influenza. And so as I mentioned before, during pandemic years, we see about 45 to 95% of the mortality due to bacterial infections. During seasonal influenza, the reporting is a little bit spottier, but it's also lower. So depending on the study, it's been estimated to be anywhere from 2 to 35%. And there's probably something to that where seasonal influenza is not quite as severe. And so the more severe of an infection you have, the more at risk you are to developing a bacterial co-infection. And the two top pathogens that are most seen are Streptococcus pneumoniae, which is a lot of what my lab works on, and Staph aureus. And we're starting to see quite a few more staph co-infections because of the antibiotic resistance with that pathogen increasing. So these are just some characteristic viral and bacterial loads where we gave mice bacterial infection at day 7. You see a little bit of an increase in the virus, which does seem to start to clear again, and the bacteria just grow rapidly. So for doses that are significantly lower than whatever would cause disease, the bacteria is able to grow very rapidly, sort of maxes out even within one day, and the animals don't clear it. So this image over here on the right-hand side is a bioluminescent bacteria, so we can image using that bioluminescence. Now there's a lot better techniques that we could actually probably see it a little bit better and in higher resolution, but it still does allow us to see the density of bacteria. And then I'm going to show these images of these lung slices, which is more of a histologic approach, looking at virus infection within the lungs. So the fortunate thing about influenza and pneumococcal co-infections is that the mouse models are very good. They follow much of the clinical features that we see. And 20 years ago now, my mentor's lab did a lot of work in characterizing this animal infection, and what they found, and what a lot of people have shown since then for not just this particular co-infection, but for other viral bacterial co-infections as well as viral co-infections, is that the order of infection matters and the timing of the infection matters. So in this experiment, if you gave pneumococcus first, you actually did a little bit better than if you had, a mildly lethal influenza infection. With them two individually, they may be a little bit of mortality. Them two at the same time, you got even a little bit more. But the most synergistic effect was when you gave influenza first, followed by pneumococcus. And when you do that, you get very rapid progression to pneumonia. So now you can see at 24 and 48 how much bacteria is in each of these mice, and it looks a lot like those bacterial titers that I just showed you. So as I mentioned, that the timing matters as well, where a lot of this work has shown that day 7 is like this really critical time when bacterial infections are the most severe. They are still lethal at other time points, but at day 7, it's much more rapid. And so a lot of the work that was done after that study had come out was focused just on day 7 co-infections and trying to understand why this is the optimal time. And I'll answer that question for you in a minute. And again, here's these data showing what the viral and bacterial titers look like when you do give the bacteria at day 7 of an influenza infection. So when I was a graduate student, so this feels like ages ago now, I started modeling these things. So this is actually like way back in the day before very many people were modeling influenza. Now there's lots of people that have done it and lots of other viruses. And the model that we use was developed in part for influenza, but also works for a lot of other viruses. It's a very simple model where we call it a target cell limited model because there's no generation of new target cells. When you do include that, you can get chronic type of infections, which of course we don't see with influenza so much. But it's a nice way to model it very simply, where virus infects these target cells, which are the epithelial cells mostly. Those cells then will produce virus. That virus is cleared out, but what we include here is just non-specific mechanisms. And then the infected cells are cleared out, which in this baby model is also not saying anything about the mechanisms by which that's happening. And we get pretty good fits of this model to the data. We've since updated this because we had some differing dynamics, which I'll show you in a couple slides. Now, bacteria, on the other hand, are much more difficult to model, and that's in part because you can't approximate their dynamics by saying that they infect cells and those cells produce more of them. Pneumococcus doesn't need a cell to reproduce. It can just sort of do that on its own as long as it has, enough food around to do that. And so its dynamics are much more tied to the host response. And so when I built this model, I sort of did it in stages where I looked at the immune responses that are happening in the very beginning, which one of them is the alveolar macrophages. So those are your cells that are supposed to keep your lungs protected from any foreign thing that is coming in. If you only include those cells, you can get a nice dose response here. So just depending on how much bacteria you're giving, you can either get growth or potentially clearance. We did add in other immune responses, including the neutrophils and the inflammatory macrophages, just to show you that we have a model that does a pretty good job of capturing these dynamics, and that it's required to have these other responses to either get clearance or at least get a little bit of clearance. In the higher doses, we don't actually see resolution of the infection. But for the purposes of the co-infection, we sort of stripped away that because it was a lot of complexity that we didn't know how to model for influenza yet, and only looked at that alveolar macrophage response to answer questions about susceptibility, which might be different than answering questions about pathogenicity. And when we did that, we went into this model where we only have these alveolar macrophage clearance added in with the bacteria and with the virus equations, and went in through each one of these different processes and asked, okay, if we alter this, can we replicate the dynamics? And what this modeling venture showed to us is that we could, but we only needed 2 mechanisms in there, and those mechanisms were completely independent. And so one of them was a suggestion that the virus somehow decreases the ability of the alveolar macrophages to clear out bacteria. And this decrease was about 85%, so it was pretty robust. The functional form that we used was a nonlinear form, so we're still not saying anything about mechanisms necessarily, just that the virus somehow does this. And then the second part of what was needed in the model to reproduce this increase in virus that we observed was an increase in virus replication because of the presence of the bacteria. And again, we're not saying how this happens because we didn't know how that happened, or at least we didn't know at the time how that happened. And again, there's a nonlinear equation here, which probably suggests there's some intermediate mechanisms. So since then, a handful of studies have either looked at this directly or indirectly and provided some support for it. One of them came out of my mentor's lab, where he had a graduate student who looked further into the alveolar macrophage dynamics. So surprisingly, at the time, nobody had looked at the dynamics of alveolar macrophages, and it was actually still even questionable if we were using the right antibodies to stain for them to allow us to understand whether or not they were alveolar macrophages. And so what he used was this labeling dye so that you could actually track the cell and make sure that the cell that was, there in the beginning was actually cells that were there later on, or if they were being depleted out. And what his experiment showed is that throughout the course of the influenza infection, alveolar macrophages are depleted. And so you can see the depletion kinetics up here on the right-hand side. And at day 7, they are depleted maximally, and their depletion is about 85 to 95%. And then after that, you can see the recapitulation of the population back to baseline after, I don't know, a week or so. So this told us not only that there was something going on there, where during the virus infection, they were impacting the alveolar macrophage response, which our model suggested, but it also verified that we were probably capturing the percent right, because our parameter that was controlling that came out to be estimated at about 85 to 95%. And from a mathematical perspective, this is also interesting because the parameter was correlated to one of the other parameters. And so the fact that we can still get accurate parameter values in the face of correlations, I think was really important and interesting, and gave us some insight into the underlying mechanism that we were actually modeling. So probably a long story short is that we used this nonlinear equation, which was probably completely wrong, but allowed us to still capture the mechanism that was at play, as well as the parameter value that was driving that those dynamics at least at that time point. So we followed this up now that we knew what that actually meant. We went back to the model, did some analysis to find a bifurcation, which is this dashed blue line here, that suggests that the percent of alveolar macrophage depletion and the amount of bacteria both matter in terms of dictating whether or not bacteria was going to decrease or increase. So if you have combinations that are below this, you get decreases in bacteria here, which you can see nicely on the right-hand side. And if you're above it, you get increases in the bacteria as the increase happens in depletion. So we had taken this idea back into the lab several years ago now. This is just a subset of that data. We did maybe more than 20 different scenarios to try to understand whether or not this bifurcation was accurate. Right here, we're looking at just a day 5 of an influenza infection, which is about a little over 60% depletion. At different doses of the bacteria, you can see whether or not bacteria clear or grow. And as the closer you get to that threshold, you sort of switch from this clearance phenotype to a growth phenotype. And the further you get away, the faster and faster the bacteria are able to grow. So that also told us that we're probably, at least from a modeling perspective, capturing those dynamics right, and that this bifurcation is really real and can, at least for the first part of the infection, dictate which way bacterial titers are going to go. But the second half of this mechanism, because it's a completely independent mechanism, all those experiments didn't tell us anything about what our model suggested about increases in virus replication. But a couple other studies have so far, and one suggested that staph, which is a separate pathogen, so there is a question about whether pneumococcus can do this, which is what our data used, but staph, when it's co-infected, can actually get inside of an influenza infected cell, down-regulate the interferon responses, which are sort of trying to keep the virus at bay and from replicating. And when it releases that suppression, you get more virus replication. So that gave us probably like a good idea that we're maybe not totally far off, and that might be the underlying mechanism that's driving this. Another set of studies showed that there was some interaction with the neuraminidase. So influenza uses neuraminidase to cleave itself away from a cell. This is why antivirals work well, because they're neuraminidase inhibitors. But when you put the bacteria in there, this bacteria in particular has neuraminidase of its own, and it can use those to help cleave off influenza, and you can actually get rescued influenza replication even in the face of an antiviral. So this obviously makes antivirals slightly more problematic if you're thinking about them in the context of somebody having a co-infection. But you can understand how that might lead to increases in virus replication. Another study showed that pneumococcus, once it's there during the co-infection, decreases the size of the influenza-specific CD8 T cell response. So CD8 T cells are there to clear out your virus-infected cells. So you could imagine if they're decreased, you're not going to get clearance of the virus. We did an experiment where we depleted out CD8 T cells. And this is just during the influenza infection. So we went in before the infection, at the time of infection at day 3 and 7 to deplete out these cells. So you can see how much lower they are in the black compared to the magenta. So we get a significant reduction in CD8 T cells. Later on in the infection, that does lead to slower clearance, but it does not lead to increases in virus replication. So while we think this might contribute a little bit, it doesn't explain why we get an increase in virus and not just a delayed clearance of virus. And then more recently, another lab here in Memphis showed that influenza can actually directly attach to bacteria, which you can imagine then that by attaching to it, it can get to new places. And this is sort of a hypothesis that we're working on. I'm going to show this image of this lung here in a minute again, but it's feasible that if the virus attaches to the bacteria and the bacteria can more freely get to different places in the lung, that you might get new areas of the lung becoming infected. So to try to answer some of these questions a little bit more, but also answer questions about whether or not our model can replicate some of the dynamics that are happening in the lung, we took an approach that we call quantitative histomorphometry. So what you're looking at here is a whole lung slice that then was stained with influenza NP protein. And so when you do that, you're essentially staining a cell that's infected with the influenza virus because they express NP on the outside of their cell. So you can see all the darker places here, that's the antibody staining. And so that's where the influenza infection is. We wanted to do this quantitatively for obvious reasons. And so we had the pathologist draw the area of the lung, which is outlined here in green, and then get the area of the infection. So all the infection is outlined in red. And if you compare the two, you can get an estimate of the percent of the lung that has been infected with influenza. So when we look at that over time, we see, of course, increases in the amount of the lung that becomes infected. And if you want to see the earlier time points, it's in this eLife paper. By about day 6 or 7, you do start to see clearance. And then by day 8, when virus is almost gone, you see almost nothing. There's probably a few infected cells here and there, but not so much for that a pathologist is going to see them from this kind of zoomed out view. And if you calculate it out, you can't see the difference so much between 8, 9, and then we went out to 10. You do start to see resolution of the damaged areas in the lungs starting to happen. But if we quantify this out, you can see that here on the bottom in red. So you get this increase in the percent of the lung that is infected. In these animals, that got up to about 35 to 40% of the lung becoming infected. And then once the CD8 T cells are there, what we call an active infection goes down to almost nothing, which corresponds to virus actually being completely cleared out. So in our model, if we take the cumulative area under the curve of the infected cells, we can get an estimate of this. So the left-hand side is a fit of the model to the viral loads. And this is where I said we had to update the model because we have a nice nonlinearity in here. And we did include CD8 T cells within this model. But the right-hand side is really just an overlay. It's not a fit to these data. So it's just an overlay of what our model predicted the cumulative area under the curve of the infected cells against the active lesion. And we do a pretty good job of being able to capture those dynamics. So without modeling anything spatially and not even taking into consideration spatial aspects of what's happening in the lung, we still can get a pretty good idea of how much of the lung is becoming infected with influenza. So we took the same approach for the co-infection. So we already knew that viral loads increased. So one idea is then that more of the lung is going to become infected, or vice versa. We could say maybe not more of the lung is becoming infected, but the replication is just increased, and we might be measuring it more. And so what this experiment showed is actually the former, where more of the lung is becoming infected with influenza. So where we see higher viral loads, and in these experiments, we did a co-infection at day 3, day 3, 5, or 7, and you get an increase in virus no matter what time you do the co-infection at. And you can see that that corresponds nicely to what we see in these images, where we get this huge increase in the amount of the lung that has become infected. And even at this day 5 co-infection, where it's an influenza infected animal might be clearing it, the co-infection is obviously not clearing it at all, and quite a bit of the lung is infected. And then at day 7, it really nicely, I think, shows it, because here, a lot of the lung has already resolved the infection, but there's still enough virus around that you actually get it in new parts of the lung, and you can get an increase in virus replication. So with our model, because we use that target cell limited approach and all our target cells were already taken up, what we did was tested out whether or not we could just increase them. Again, as sort of a way to say that this is just a caveat of the model, and we just let's see if we can add in, new target cells that it suddenly has access to, and that does seem to work, where we get this increase in the amount of virus just by having new cells to infect, and those cells are still going to produce virus, but produce it at the same rate that it was beforehand. And again, if we take the cumulative area under the curve now of the infected cell dynamics, we actually do still capture this increase in the area of the influenza infection. And what these data show then, now quantified out, is that at this day 3 co-infection, you get nearly 80% of the lung becoming infected with virus. And as you are sort of more in a clearance realm, where you're out at day 5 to 7, where the CD8 T cells have shown up and are starting to clear out virus, it's a little bit lower, but it's still measurable as far as how much more of the lung is becoming infected with the virus. So this experiment also showed us that alveolar macrophages, oops, getting ahead of myself there, that alveolar macrophages are decreased even further during the co-infection. And so one hypothesis would be asking whether or not this increase in virus just leads to more alveolar macrophage depletion, since the alveolar macrophages are depleted out in some way by the virus anyways, or is it them trying to kill bacteria and then their population decreasing because they're doing that. So now this is what extra data always does for this, is open up new questions that we sometimes dive into. This was one of them, but we wanted to do it at least first from a mathematical perspective. And we already knew we needed to update our model, because our model had a couple things in it that or maybe approximations in terms of those nonlinear functions, or as I mentioned with the target cell limited aspect of this model, we had to add in those additional target cells. But because now we know that the alveolar macrophages actually have dynamics, we wanted to then model those dynamics. And that was not as straightforward as one would hope. And what we found in this work is that it required an intermediate. And so what we couldn't do was capture anything about the alveolar macrophage dynamics with populations that were already in the model. Instead, we thought, okay, well, maybe it's not directly from the infected cells or the virus, and there is some intermediate that's just not in our model. And so we took that approach, and instead of trying to think of what that might be, we said, okay, there's an intermediate. Let's find out the dynamics of that intermediate that could allow us to capture the alveolar macrophages. And that the dynamics are over here on the right-hand side of what this intermediate variable would need to be to cause this depletion in the alveolar macrophages. So sort of in the midst of us doing that, this paper came out looking more at how influenza induces the alveolar macrophage depletion or dysfunction and allows pneumococcus to grow rapidly, suggested that interferon gamma was one of the drivers. And so we thought, okay, let's just plot our intermediate dynamics against our data for interferon gamma. And you can see that here. This is what we might have predicted those dynamics to look like. It's not a perfect fit by any means, because it's actually not a fit at all, but it does overlay pretty nicely and mostly captures a lot of the dynamics. And so we're following this up now to try to get a better model for interferon gamma and its regulation of the alveolar macrophage response. So hopefully in the next couple months, we'll be able to finish up that work. But if we put these things together and look at seeing if we can model each of the co-infections without changing any parameter values, the short answer is we can. So we can capture the viral, bacterial, and alveolar macrophage dynamics just by changing, the day of infection, and that's pretty much it. I did include in this work 2 realizations for the co-infection at day 3, and that's in part because you're sort of on that bifurcation, and that there's probably other dynamics that are playing into the large heterogeneity that we see when we're close to that threshold. And you can see that here where we have a pretty good span of bacterial dynamics depending on the animal, where some clear it and some don't. And just by changing little tweaks to parameter values where you might get a tiny bit more virus with actually not even measurable increases in alveolar macrophage depletion, you can still get pretty different dynamics for the bacterial dynamic, or for the bacterial loads. And that's in part because of that regulation around that bifurcation. And then as you get sort of further away from that, as you are getting more and more depletion, that heterogeneity goes away. And that's true biological heterogeneity that we can replicate. But fitting this model to the data suggested that the alveolar macrophage depletion that happens during the co-infection is probably still mediated by that increase in virus. But we definitely need to look at that a little bit further. So that gave us at least some indication that the 2 mechanisms that we found in that early work were probably true, but also had a lot of underlying dynamics that we didn't know about. But we also know that both viral and bacterial loads are not directly connected to severity. And often disease severity is more connected to host immune responses or to what people call inflammation. And different people define inflammation in different ways. One is from maybe a more pathologic standpoint, where say a pathologist might look at a tissue section and define inflammation that way. That's what these data here are. So the scoring is 1 to 5, and the yellow and the white are interstitial and alveolar inflammation, which for an influenza infection are relatively similar. We decided to try to develop a little baby model that was just proportional to the infected cells, with the idea that infected cells being present brings in cytokines and cells that ultimately cause inflammation. And it worked it works pretty well, at least for the influenza infection. And if we look at whether or not inflammation is actually connected to disease severity, as well as the amount of the lung that has been infected by influenza, what we see here is that it probably is, but it's nonlinear. So we just used a nonlinear correlation to relate these two things, and splitting alveolar and interstitial inflammation here. They were, their dynamics are similar, so there wasn't a lot of difference there. And it probably correlates quite a bit more to interstitial and alveolar inflammation than it does to the amount of the lung that is infected, but not so much on sort of the nonlinear upswing, where you get a pretty tight correlation between the two. But this data also suggested that just because you have inflammation, or just because you have a large amount of your lung becoming infected with influenza, does not necessarily equate to increased disease severity. So you can have no increases in these things, and your disease severity can be continuing to get worse. And so there's probably, because it is nonlinear, there's probably other mechanisms that are happening there that we need to think about, at least as far as interpreting the data. But we, of course, extended this to the co-infection, and this was actually an instance where our model completely broke down. Because we have a too simplistic of a model, the increases in infected cells might suggest increases in inflammation, and that's not actually what we see here. And we didn't see a whole lot of increased inflammation in the co-infection, even with things increasing that are typically associated with inflammation. And two of those things are macrophages and neutrophils. If you just plot these two things against each other, you do see a pretty good log linear correlation between them that holds true. So the white compared to the colored circles and triangles is the influenza versus the co-infection. It's probably still true that there's a log linear relationship with inflammatory macrophages, and potentially with neutrophils as well, where it was nicely log linear. But in the co-infection, there's a large neutrophilic response, and neutrophils are typically associated with inflammation, and there is a slight increase here, but it's not necessarily what one would have expected. So you're not getting a large increase in inflammation while you do have this huge neutrophil response. And so we probably need to follow up on trying to connect these things a little bit better. The story about influenza co-infections is much more complex, where over the last 20 years or so, people have shown lots of mechanisms and a lot of things to be different during the co-infection. The question, at least for us mathematically, is, do these things matter, or do they contribute to the dynamics, or are they connected in some way? So some of these things are, say, increases in bacterial attachment, increases in receptors. There's, of course, decreases that happen in just mechanical clearance of the pathogens. Cytokines either are significantly up or down. Some of the cell types are impaired. So I talked a lot about alveolar macrophages and T cells, but neutrophils also have impaired function, as well as NK cells. So they might be contributing to some of these dynamics. And so we probably have a lot to follow up with mathematically, but from a larger perspective, this more macro type of model did a pretty good job of identifying at least a couple of the main mechanisms that were driving the dynamics that we were seeing. So in the last few minutes, I'll talk about our work on SARS-CoV-2 and whether or not it increases bacterial susceptibility. So we obviously wanted to look at this during the pandemic because of what we knew about influenza and what we know about some other respiratory viruses being able to enhance susceptibility to bacterial pathogens. And there were a few studies that were coming out early on suggesting that some patients did have bacterial or fungal co-infections. So we went at this from an experimental perspective, where we used the same experimental design that we had used previously, although we're in a different animal model here, because at that point, the only animal model available was these K18H2 transgenic mice. Now there's adapted strains that we can use other mice with. So there's lots of questions still about these animals. We had no idea how a bacterial infection was going to behave in it, so we just sort of went at it blindly. We also used a low dose of SARS-CoV-2 because we wanted to give the animals the best chance of survival so that we could study the co-infection and whether or not it was increasing pathogenicity. And then we'd used, again, the day 3, 5, and 7 or 7, not and 7, to introduce the bacteria. And what these data show is that the secondary bacterial co-infection does increase lethality, as well as morbidity as terms of weight loss, but it's not quite as robust as it was with influenza. So at day 3, we actually saw almost nothing. There was no increased lethality. Weight loss was maybe slightly altered. But as you get towards day 5 or 7, you started to see these increases, both in lethality and in weight loss. But we did have some mice live and survive the co-infection, which we hardly ever, if ever, see with influenza. It's pretty much 100% lethal infection. But some of the dynamics were completely distinct from what they were with influenza. So we did see increased bacteria. These data probably suggest that maybe these K18 mice have a different threshold, because at day 3, we saw almost complete clearance of the bacteria. At day 5 co-infection, it's probably sitting right around that bifurcation. And then we finally get increased bacterial growth at that 7-day co-infection. We did not see it get into the blood so much. That's something I didn't show with influenza, but this bacteria gets readily into the blood. And we only saw that in a handful of animals at day 7. And with the virus, where we saw all that increase with influenza, as far as viral loads and the increased infection in the lung, we don't see that with SARS-CoV-2. So there was no increase in virus. And if you look at these histomorphometry type images, you can obviously not see a difference here, and that's because there is no difference. So that same increased dissemination that we see during influenza, we don't see during SARS-CoV-2. And you can see that a little bit with these images, where in these ones we actually stained for pneumococcus. So this is a zoomed in version of this, but it's similar slices. So these 3 little airways here, you can see pretty much the same 3 that are on this side. With SARS-CoV-2, there's a lot of cells infected, and pneumococcus is sort of in an adjacent area, so it's close, but it's really hanging out, not exactly so much on top of SARS-CoV-2, which might suggest that maybe SARS-CoV-2 doesn't attach to pneumococcus like influenza does. So that's just a hypothesis. Obviously, we would have to prove whether or not that's true, but it might explain why we don't see the increased dissemination of SARS-CoV-2, which is probably a good thing, because it might actually make the co-infection significantly worse if SARS-CoV-2 could do that. We didn't see a whole lot of changes in the immunology. We found a few here and there. So alveolar macrophages do still deplete in this animal model, but they didn't deplete more during the SARS-CoV-2 bacterial co-infection. And again, if we think back to the mechanisms during influenza, it's probably because that virus increased. And here we don't see that increase in virus, so we wouldn't expect an increase in the alveolar macrophage depletion. We did see small changes in the inflammatory macrophages, but not actually until after resolution. So as I mentioned before, not all of the animals succumbed to the infection. And so we were able to follow them out a little bit more, and we looked out at day 17. So virus is cleared and bacteria is cleared. And in theory, the lung is trying to repair itself from the infection. But what we do see here, even though we didn't see changes in macrophages early on, we did see a change in them later on as far as the lung remodeling is concerned. So there might be some interesting things there with changes that occur to the host responses during the co-infection that are maybe long lasting. And the same was true for CD8 T cells, where there's not a huge difference during the infection, but we did see a small difference in the recovery. Similarly with B cells. During the infection, one of the only cell types we did find different was neutrophils, and where they're increased during the co-infection compared to the SARS-CoV-2, but pretty much only at day 7. And only a few select cytokines were different, and not at all of the co-infection time points. So we saw minor differences at the day 5 co-infection in IL-6, IL-15, IL-18, and LIF. And then with a couple of these lower amounts, actually, so maybe even some down regulation of the cytokine response during that repair phase. And for some of these are actually even lower than what they were at in a naive animal. And so there might be some significant down regulation happening there that could be interesting to follow up on. A couple other cytokines that were related to those changes in macrophages and macrophages and CD8 T cells out at this day 17 time point. You see their associated cytokines with MIP2 alpha, which is a macrophage related cytokine. So macrophages were increased. You see an increase in that particular cytokine. Same with CXCL10, and then RANTES, which is a CD8 T cell related cytokine. And so in summary, what we know so far is that bacteria can significantly increase pathogenicity during influenza and maybe to a lesser extent to SARS-CoV-2, but it is still present. There are similar features between the two pathogens, but as I mentioned with the dissemination of the virus and maybe the alveolar macrophages, there are some distinct features as well. So I'll leave you with my thoughts on why we saw so few co-infections during the COVID pandemic. And one is that there was a lot of physical distancing and mask wearing. And so a lot of bacterial pathogens, as well as other viral pathogens, weren't being transmitted quite as much. And so there was actually very little community-acquired pneumonia that was not SARS related. There was also low testing and reporting of the bacterial infections and prophylactic use of antibiotics, where some studies suggested at least in 70% of patients they were given some sort of antibiotic, which probably helped decrease their chances of acquiring a bacterial infection. So we may see more going forward now that we're not physical distancing so much and not wearing masks, and that children are back in school. But from what our data show, at least, it might still be significantly less than what we see with influenza, which probably is a good thing. So I will stop there and take any questions that you might have.
Hi, Amber. Thanks so much. That was such a great talk. It's really nice to see like the merging of math and experiments. I had a quick question, then I'll throw it out to the rest of people as they get them ready. But I was wondering, because you see different dynamics in the influenza and SARS-CoV-2 infections, do you think that there's an effect of the timing of the co-infection in the animal model?
I do. Retrospectively, probably choosing day 3 instead of 10 or something was probably an oversight. Well, we didn't know what time points to go into, but I could imagine that we might see a little bit more pathogenicity if we looked later on, given that SARS-CoV-2 doesn't peak until day 5-ish. And has longer lasting effects. So even when we do see virus pretty much being cleared by day 7 or 8 at least in the animal model, the effects of it last longer than that. And so yeah, we probably, like I say, retrospectively, we probably won't do it now. Went in at day 10 or 14 or something and probably saw, gosh, we could have went in at 17, given that there's still immunologic differences
Yeah. And do you think that that maybe is in some sense, like you mentioned, the binding of the viruses to the bacteria, but do you think that also that in some way supports, or I guess, I don't know, what are your thoughts about what that means about the interferon response? Because that was one of the things that was mentioned, you know, in terms of the influenza severity.
Yeah, that's a good question. Oh gosh, I don't know. I mean, depending on what mechanism we're talking about might depend on like what time scale we're on too.
Mm.
So of course, interferon is going to happen a little bit earlier on, where maybe some of these other effects, say like on neutrophils, might happen a little bit later. During the influenza bacterial co-infection, neutrophils play a large role, and that has we haven't addressed. So we've addressed it experimentally, but we haven't addressed it mathematically so much yet. But we have some other data that show that if neutrophils are sort of happier and behaving better, that there's less pathogenicity and less lethality. And the fact that neutrophils are so impacted by SARS-CoV-2 might say that it would have effects later on.
Right. Yeah, that makes sense. Okay, I have other questions, but if I'm going to open up the floor, otherwise I'm going to monopolize the time. So I don't know if somebody else has another question, you can write in the chat or just unmute yourselves.
It's Michael. I have perhaps a really naive question. At a couple points, you said that the nonlinearity that you were finding suggested that there was some intermediate step. I don't understand all the microbiology that you were talking about, but could you give a bit of intuition on how you made that connection?
Yeah, I always view nonlinearities to be probably one of two things. Either it's a true nonlinearity in the way that we think about, like, you know, Michaelis-Menten and the impact of, you know, two things coming together in different quantities can actually change the dynamics. In immunology, I would say there's probably some of that, but because it's so complex, the nonlinearities to me also sometimes suggest that there probably is an intermediate, that it's a good approximation for how we're modeling it, but it's probably not exactly the mechanism. And that was highlighted for us in that co-infection work, because we had a nonlinear equation, but you needed parameter value differences if you had co-infections at different times. So for our initial work, we did it at day 7. But if you use that same function without changing anything for a day 5 co-infection, it didn't work. Like, once we had the data, we found that it didn't work. And that was probably because not only parameters might be different, but that nonlinearity did not capture the dynamics of that depletion of the cells properly. So it's almost sometimes all the data that we have to deal with, that we're looking at, might not have all the information that we need. And so we use nonlinearity sort of as an approximation for that. And like I said, what we've found so far is that those nonlinearities are usually sort of underlying mechanisms that we're not actually modeling directly.
Okay.
Sorry, go ahead, Michael. Sorry.
Oh, it implicitly then there's a strong assumption that if things are linear, then there's no complicated underlying mechanism. But
yeah, maybe.
But in all sorts of other things, logistic relationships, for example, or exponential decay or whatever are more typical. They are natural.
Yeah, you make a good point with the exponential decay, because we can model virus decline with an exponential decay. But there is a mechanism that we're not modeling by doing that, right? Which is the CD8 T cells. And so, yeah, I don't know, that does make it, I don't know, more complicated and maybe not totally direct to say that only nonlinearities do that, because even if you model something with a linear type of term, there could still be other mechanisms that drive that. That's what makes it hard.
Thanks, very interesting.
So there was one question in the chat from Mohammed, and he said, it was noticed that repeated exposure to influenza reduces the immune response. Could we expect this, for COVID-19 as well?
Yeah, that's a good question. I would say probably. So there's memory that occurs. Usually we think about memory in terms of CD8 T cells, but there's a lot of new work suggesting that even some innate cells have some sort of memory. But also there's adaptations that occur. So there's of course adaptations that occur on the virus side where we start to see mutations happening. But there is some work that has been done suggesting that host responses and even like host tissue responses, so not even just the immunology, can adapt over time. And that might happen with repeated exposures. So it wouldn't surprise me if we do see something similar with SARS-CoV-2, where not only is the pathogenicity going to decrease, but we generally have better host responses, as well as maybe even physiologic changes to the cells because of repeated exposure. So I guess only time will tell, but it would not surprise me if that's what
I think, have you seen, I think it's Rustam who talks about this, about the path to endemicity, and I can't say it, be becoming endemic. And part of it also is related to like acquired memory through children, the exposure of children in the population, and that attenuates the immune response. So I guess, these are all kind of related in some sense.
Yeah, I think so. There's another word that has been popping up in the immunology literature called imprinting.
Right.
Yep, and I think that gets at that. That's a big question, and a hard one, I think, to address. But it does look like even over time, I think there are some reports that have even looked back at, say, like the plague outbreaks and how much that altered not even just our responses to, say, other pathogens, but even to other diseases like diabetes, for example. Right. And yeah, that's who knows, like, the short-term impacts versus the like significantly long-term, centuries from now type of impacts.
Yeah, totally. Or decades, right? Like,
yeah,
I've been saying like shingles, we probably didn't know about shingles. So yeah. Okay, well, we've reached time and we won't take any more of your time. So thank you so much, Amber, for your great talk. It was great to have you. And we'll, I guess, I don't know if there's another seminar of this year, but hopefully